Hello everyone. In this video, we are going to discuss some of the past paper questions from Unit 5 Enzymes. Okay, so let's begin. First question is objective type question from paper number 1, and the question is what are characteristics of enzymes? Fine. So here 4 options are given and you have to choose the best option which would be the correct answer of the question which is asked. So what are the characteristics of enzyme? They are carbohydrates and biological catalyst. So they have mentioned here they are carbohydrates, so this gets wrong because enzymes are actually proteins. Okay, they are not carbohydrates. This biological catalyst is correct. Then they are carbohydrates and chemical messengers. These entire option is wrong. They are not carbohydrates and they are not chemical messengers. Hormones are called chemical messengers. Fine. The third option they have given is option C is they are proteins and biological catalyst. So they both are correct, proteins and biological catalyst. Fine. And the last option is they are proteins and chemical messengers. So this option is wrong. So the correct option is option C, proteins and biological catalyst. The second question is two enzyme controlled reactions are shown. Okay. So amino acids with the help of enzyme 1 are converted into proteins. Fine. And here the second enzyme controlled reaction is proteins with the help of enzyme 2 are converted into amino acids. Now from these two reactions, what deductions can be made about an enzyme? Enzyme 1 has been changed to enzyme 2. No, this is wrong. Okay, enzyme 1 and enzyme 2 are completely two different enzymes. Okay, it's not like that enzyme 1 when it is involved in first reaction, then it will get changed into enzyme 2 for completing the second reaction. That's wrong. These two enzymes are completely two different enzymes. Okay, then enzyme 2 slows down the production of amino acid. This option is also wrong because nowhere in this particular question any data is given which can prove, okay, or which can show that enzyme 2 slows down the production of amino acids. Okay, actually these enzymes, they always increases the rate of any chemical reaction. Fine, they never slows down. Okay, they can only increase the rate of chemical reaction. Then third option is enzymes can build up large molecules. Okay, this could be the correct answer, but let's go on the fourth option also. Enzymes only break down large molecules. Okay, now out of these two, the correct answer for this question would be option C. Why? Because here they are using, they are saying enzymes can build up large molecules. This option is correct, obviously, because enzymes are actually involved in, two types of reaction. In one type of reaction, they build up large molecules. Okay, they join monomers, like for example amino acids are the monomers of proteins, so they join many number of amino acids with each other and they form one large molecule called protein from amino acids. Okay, but the option D is given, enzymes only break down large molecules. Okay, so this option gets wrong because of this word only. Okay, because enzymes can break down large molecules as well as enzymes can also build up large molecules. Okay, it can not only break down large molecules, but it can also build up large molecules. So the correct answer is option C. Third question is, an experiment was carried out to investigate the effect of pH on enzyme action. Okay, so in order to investigate the effect of pH on enzyme action, an experiment was carried out. The graph shows the result. Okay, so this is x-axis, this is y-axis. Now you have to explain, or you have to choose the option which shows the correct labels for x-axis and y-axis. Now if you read the question again, it's given here, the experiment is about investigating the effect of pH on enzyme action. Now always remember that whenever we talk about graph, whenever we talk about plotting of graph, on x-axis we always label independent variable. Okay, and on y-axis we always label dependent variable. Fine. So in this experiment, that is the effect of pH on enzyme action, pH is our independent variable. Okay, what is independent and dependent variable? I'll explain you in the next question. and the rate of reaction. Okay, the rate of reaction becomes dependent variable. Okay, rate of reaction. So here the correct answer would be x-axis would be pH and y-axis would be rate of reaction. So the correct answer is A. Remember that always we label dependent variable on y-axis and independent variable on x-axis. This point is very important as per your IGCSE exam, especially in paper number 6, okay, while plotting graph. Okay, so always 1 mark is awarded for a correct orientation and labeling of the axis. Fine. If this first step goes wrong, okay, labeling of axis, if this first step goes wrong, that is orientation and labeling of axis, then no other marks will be awarded. Your entire graph gets wrong. And the fourth question is from your paper number 6, which is ATP. ATP means alternative to practicals. Okay, so let's begin. Catalase is an enzyme found in plant and animal cells. Okay, I've taught you about catalase in previous 2 videos. I've told you that catalase is the enzyme which is found in plant and animal cells. It has function of breaking down hydrogen peroxide, a toxic waste product of the metabolic processes. So all the living cells, they have this catalase enzyme. In metabolic reactions, a toxic waste product is produced, which is called hydrogen peroxide. Now this hydrogen peroxide is highly toxic or poisonous for the cells. Hence what happens the moment this hydrogen peroxide is produced, immediately catalase enzyme will break down this hydrogen peroxide into hydrogen and it will break down hydrogen peroxide into oxygen and water. Okay. The first question is, state the term used to describe the removal of waste products of metabolism. Now always whenever you read the question, check the number of marks the question has been awarded. Okay, so here the question is asked for 1 mark. So you just have to state the term, and in unit 1 you have studied that removal of waste products of metabolism from the body of living organism is called excretion. So the correct answer for this is excretion. Okay, the correct answer for A1 is excretion. Second question is, define the term enzyme. Now this question here, if you check the marks, it is asked for 2 marks. Okay, so you have to write down 2 valid points about enzymes. I've already taught you in enzyme unit that whenever this definition of enzyme is asked for 2 marks, if you just write down biocatalyst, you will get 2 marks. Okay, if you just write down one sentence here that enzymes are biocatalyst, you will get 2 marks. Why? Because in the word biocatalyst, the word bio represents living organisms and catalyst represents anything which increases the rate of a chemical reaction. Okay, so you can simply write down that enzymes are biocatalyst, you will get 2 marks. And for example, if you don't remember the term biocatalyst, then you can just write down enzymes are made up of proteins. Okay, and they actually help in increasing the rate of a chemical reaction. Fine, they are produced by living cells. Okay, so any of these points you can write. Fine. Further in this question, it's given an investigation was carried out to study the effect of pH on catalyst. Okay, now read the question carefully. The investigation is about to study the effect of pH on catalase. Okay, effect of pH on catalase using pieces of potato as a source of enzyme. Okay, so the source of enzyme is potato. Okay, pieces of potato. Now oxygen is formed when catalase breaks down hydrogen peroxide as shown in the equation. Okay, so when hydrogen peroxide is broken down by using the enzyme catalase, the products formed are water and oxygen. Okay, the rate of reaction can be found by measuring how long it takes for 10 centimeter cube oxygen to be collected. Now this is important. Here the investigation is carried out to study the effect of pH on catalase enzyme. Okay, effect of pH on catalase enzyme, and the source of catalase enzyme is pieces of potato. Fine, and you know that once catalase enzyme acts on hydrogen peroxide, it will break down hydrogen peroxide into water plus oxygen. Okay, and how the rate of reaction is being measured? Rate of reaction is being measured by measuring or by actually checking the amount of oxygen produced. Okay, so as per this question, the rate of reaction can be found by measuring the time which is takes the time taken for collecting 10 centimeter cube of oxygen in the experiment. Okay. Now here the first question given in B is state independent variable in this investigation. Now always remember that whenever an experiment is carried out, there are two types of variables, dependent and independent. Okay. Independent variable is actually, the value of independent variable actually remains unchanged. Okay, the value of independent variable does not depend on any other variables present in the experiment. Okay, its value will not change. If there is any change in dependent variable, the value of independent variable will not change as per that. Okay, but dependent variables, they are always dependent on independent variable. Okay, for example, in this experiment, the independent variable, there is only one independent variable, and that's pH. Okay, so the independent variable in this experiment is pH. Okay, and the dependent variable is rate of reaction. Okay, now and how we are measuring rate of reaction here? We are measuring rate of reaction by measuring the time taken for collecting 10 centimeter cube of oxygen. So the rate of reaction will change as the pH will change. Okay, understand, the rate of reaction is dependent variable, and this dependent variable is dependent on pH. Okay, because as pH is changing, the rate of reaction will also change. You have studied in theory, right? As pH will change, the rate of reaction will also change. Similarly, as temperature will change, the rate of reaction will also change in our experiments. Okay, so pH, temperature, etc., they are all your independent variables. So here independent variable is pH. Suggest two factors that would need to be kept constant in this investigation. So what are the factors when you read this entire experiment? What are the factors that would be kept constant? Okay, so one factor, very important factor, could be temperature. Fine, one factor could be temperature. Then here it's shown that the source of enzyme is potato. Okay, so you can write down the constant factor could be like the amount of potato or the amount of enzyme is also fine. Okay, then concentration of hydrogen peroxide, this could be concentration of hydrogen peroxide or amount of potato. Okay, concentration of enzyme, all these things could be the two factors that could be kept constant in this investigation. Next, they have given table 4.1 shows the results of the investigation, but it is incomplete. Now here table 4.1 is shown, and they show the result of investigation. pH is given. pH is independent variable. So they have actually collected data over the pH from 4 to 8. Okay, between pH 4 to 8. So pH 4, 5, 6, 7, and 8. Here they have given time to collect 10 centimeter cube of oxygen per minute. Okay, so it is per minute, 10 centimeter cube of oxygen per minute. So at pH 4, 20 minutes were taken. Okay, so at pH 4, 20 minutes were taken to collect 10 centimeter cube of oxygen. At pH 5, the time taken was 12.5 minutes to collect 10 centimeter cube of oxygen, and similarly goes down. At pH 7, 13.6 minutes were taken to collect 10 centimeter cube of oxygen, and at pH 8, now you have to calculate how much. Okay, at pH 8, 17.4 minutes were taken to collect 10 centimeter cube of oxygen. Now in the third column, rate of oxygen production is given, and the unit of rate of oxygen production is centimeter cube per minute. Okay, in one of the video I've taught you rules to draw the table. Remember that whenever we draw table, the unit of any quantity is always written in the heading column. Okay, you can never write a unit along the body of the column. Okay, remember this. that you can never write unit along the body of the column. Okay, unit is always written in the heading cell. Okay, so wherever the heading of the quantity is written in table, unit should always be written along with the heading of the quantity given in a particular cell. Okay, unit can never be written along the body of the column. Okay, so when you write this centimeter cube per minute, if you write here in every cell along the body of the column, you won't be awarded marks. The table will get rejected. Okay, remember that. So in third column here, rate of oxygen production is given and the unit is centimeter cube per minute. Okay. Now the rate at pH 4 is 0.5, at 5 it is 0.8, at pH 6 it is 1, at pH 7 it is 0.74, and at pH 8 you have to calculate the rate of oxygen production. Okay. So if you look at C, the question C is calculate the rate of oxygen production at pH 8. Show your working, write your answer in table 4.1. Now here the question is actually given for 2 marks. So remember they have specifically mentioned that write your answer in table 4.1. So until and unless you won't write your answer here in table 4.1, you won't be awarded complete 2 marks. Okay, so 1 mark is given for writing your answer in table 4.1. Okay, now you have to actually calculate here rate of oxygen production at pH 8. Fine. Now the formula for calculating rate of reaction. Actually, this rate of oxygen production is nothing but rate of reaction. Okay. So if you will see the formula for rate of oxygen production or rate of reaction is, it is delta C by delta T. Now the meaning of delta C is change in concentration of product. Okay, if there is any change in concentration of product, or you can say the amount of product produced in that particular duration of time. Okay, T refers to change in time period, or just you can say time period. Okay, so in this particular time period, what is the change in concentration of product, or what was the amount of product produced? Here the product is oxygen. Okay, now we know that the amount of oxygen produced is always is given constant here. Okay, so constant here. Actually we are taking 10 centimeter cube of oxygen for every pH. Okay, so here delta C would be 10. Okay, 10 centimeter cube per minute, 10 centimeter cube. Okay, and the time given here is, it's written here at pH 8, the time to collect 10 centimeter cube of oxygen was 17.4 minutes oxygen. So as per the formula, the rate of reaction will be 10 divided by 17.4, and the correct answer you will get is 0.57 centimeter cube per minute. Now it's compulsory to write down the unit here in front of your answer. Okay, so in order to show this entire calculation, that is just 10 divided by 7.4, if you show 10 divided by 17.4, if you show and you will write down centimeter cube per minute, you will get 1 mark. Okay, and another 1 mark is for writing the answer in the table. If you will not write unit here, marks won't be awarded. So remember that whenever you solve such questions, you have to mention the unit. Okay, and as it's mentioned here, write your answer in table 4.1. So you should also write your answer in table 4.1. Hence you will get complete 2 marks. Question D is, complete the graph by plotting rate of oxygen production against pH. Okay, now here they have already given you the scale. Okay, and you know that pH is independent variable. Okay, and rate of oxygen production is dependent variable. So we know that on x-axis it's always it's always like dependent variable, that is pH, and on y-axis it's always independent variable. Okay, on y-axis it's always independent variable, that is rate of oxygen production. Now one very important point about plotting of graph is, whenever you are labeling the axis, okay, this is x-axis and this is your y-axis. So on x-axis you will label pH, okay, and on y-axis you will label rate of oxygen production. So whenever you're labeling x-axis and y-axis, make sure that you actually write down the unit of these labels. Okay, for pH we don't have any SI unit. Okay, but when we are writing here rate of oxygen production, so if you look at the table, rate of oxygen production is actually measured in the unit centimeter cube per minute, isn't it? Rate of oxygen production is given in unit centimeter cube per minute. Okay, so while labeling the axis here, rate of oxygen production, you must write down the unit centimeter cube per minute. Okay, and then you have to plot the graph. Now remember that your the pH range which is given in the table is between 4 and 8. So at pH 4, the rate of oxygen production was 0.5. At pH 5 it is 0.8. Okay, so if you look here, at pH 4 it was 0.5. So this is 0.2, this is 0.4, and here it is 0.5. Okay, at pH 5 it is 0.8. So at pH 5 it's given clearly here, it is 5, and at pH 6 it is 1. Because at pH 6 it's given here, it is 1. Similarly, if we'll go down at pH 7, look at the table once. At pH 7 the rate of oxygen production is 0.74, and at pH 8 it is 0.57. So at pH 7 it is 0.74. Okay, now this is 0.6. Okay, 6, and then 0. Here it is in the middle, 0.7, and then 0.7, 1, 7, 2, 7, 3, 7, 4. Okay, and then 7, 5, 7, 6, 7, 7, 7, 8, 7, 9, 7, 10. Okay, so this is 0.74. Okay, you actually have to choose your scale very wisely. Here actually already the scale is given. Okay, so you have to plot your graph by using this scale only. Otherwise, you have to choose your scale very wisely so that the plotting of these points on the graph becomes easy. Okay, and at 8 it is 0.57. Okay, so this is 0.4. Here it is 0.5, 0.51 and 2, 0.53 and 54, 0.56 and 57. Okay, 0.58 and 59 and 0.6. So 0.74. Okay, so this is how is the graph. Okay, you can just connect these points by using a single and clear line. Okay, now whenever you plot the points and connect these points, so remember it's at one point, the last point. After joining the last point, you should never increase, you should never increase your line beyond the last plot. Okay, this is called extrapolation. Okay, what it is called? Extrapolation. And sometimes, the marks is actually deducted for this extrapolation. Okay, so you should never increase your line beyond this particular, beyond this last point. Okay, I have also plotted the graph and I'll show you. Okay, so remember x-axis is pH and y-axis is rate of oxygen produced. Okay, and as I've told you, I've mentioned here the unit centimeter cube per minute. Okay, and here you have y-axis and here this is x-axis. Remember that on x-axis we always label independent variable, and on y-axis we always label dependent variable. Okay, and after plotting the graph, either you can draw the smooth curve which is joining all the plots as I've shown you in the upper graph, or you can just draw one single line passing from the origin. Okay, the single line passing from the origin, this is origin, the single line passing from the origin, and just passing through the center of these points, only passing through one point and from the center of these 4 points. Okay, okay, so this graph was actually very simple. Scale was already given. You were only supposed to label the axis and label these axis and then plot these points on the graph and then draw a single line between these points. Fine. I'll show you in marking scheme what it's given. The question was asked for 4 marks, plotting of the graph. So 1 mark you will get for x-axis labeling. Okay, so if you label x-axis with pH, you will get 1 mark. Second marks you'll be awarded if you will label y-axis with rate of oxygen production, or just you can write on rate of reaction. If you write on rate of reaction, that is also fine. Okay, and they have mentioned here centimeter cube per minute or centimeter cube per minute, like in any way you can write down. Okay, so understand what I mean to say is, whenever you're writing rate of reaction or rate of production, whenever you're labeling y-axis with rate of reaction or rate of production, you must mention the unit there. Okay, centimeter cube per minute. Okay, then third mark you will get if you will plot all the points correctly. Okay, so you have to plot all the points, that is at which pH, at one particular pH, how much was the amount, how much was the rate of reaction. Okay, so when you will plot all the points correctly, you will get 3 marks. Okay, for example, if there is a mistake in any one point, third marks will be awarded. If there is any mistake in plotting of 2 or 3 points, the entire 1 mark will be deducted. Okay, no mark will be awarded then for the plotting of graph. Okay, and last mark, that is 4th mark, will be awarded for continuous and clear line. Okay, which may be either a curve which may not go through all the points, or straight lines between the points. Okay, so I've given you both the options above. Okay, it can be a curve. Okay, and it can be a straight line passing between the points. And they have given here reject if line goes beyond plotted points. Okay, so if line will go beyond plotted points, you have to reject. This rejection is actually given about the curve, as I've shown you here above. This line should not go beyond the last plotted point. Okay, if it goes beyond plotted point like this, you won't get any mark for the last option, that is joining of the plot. This is called extrapolation, and extrapolation is not allowed in the line graph or IGCSE papers. Okay, so I hope I'm clear with the plotting of graph. The next question is question E. They have given 2 options, 1 and 2. Okay, now the question is, using data from the graph, describe the changes in reaction rate between pH 4 and pH 8. Okay, and second question is explain. You have to understand very well. This is a very important question. Whenever they give you this table or graph question, they ask you to describe and explain the graph and table. Okay, but there is a big difference between describe and explain. Okay, so as per your IGCSE guidelines, I'll just let you know first. Whenever a describe question is given, so describe question can be given on topics like they might ask you to describe the steps in the process. Okay, and in any process describe the steps, this question could be given, or describe the appearance of a biological structure. Okay, if any biological structure is given to you, so you have to describe that particular appearance. Okay, and you may also have to describe some data given in a table or a graph. So our question right now is based on this sentence. Okay, some data given in table or graph. Okay, make sure you have the correct vocabulary for such a description, and use the words increase, decrease, constant, peak, maximum, minimum, etc. Okay, along with this, you should also, from your table and from your graph, you should actually also quote the data. Okay, this is very important. Okay, along with all these important points, you should actually quote any kind of data from the table in order to describe your table or graph, in order to answer these describe questions. Explain is this is not the same as describe. Now understand that describe and explain, they both are different questions. In explain, actually see, whatever you have written in the describe question, okay, you have to actually explain the reason behind these describe. Okay, reason behind what your answer in describe. Okay, so in describe, you just have to you just have to write down the pattern of the graph. Okay, in describe you have to write down the pattern of the graph. You have to use quoted. You have to quote data from the graph or table. Okay, but in case of explain questions, you have to explain the reason behind such pattern of the graph. Okay, you should give an answer that has some reasons. You may have to explain why something happens or how it happens. Okay, so you have to explain that why you're getting so and so curve or so and so pattern in the graph. Okay, so you have to actually explain the reason behind getting such a pattern or curve in the graph. Okay, why something happens or how it happens. Now, if you'll see this question, the question E is using data from the graph, describe the changes in the reaction rate between pH 4 and pH 8. Okay, so if you see between pH 4 and pH 8, you have to describe the changes in the graph. Now, if you look at the points plotted, as pH increases, okay, so as pH increases, that is as pH goes from 4 to 8, the rate of reaction increases. Okay, 1 mark will get for this. But rate of reaction increases only till pH 6. If you look here, the rate of reaction increases only till pH 6, only till this particular pH. Okay, second mark. Third mark you will get, or you might get if the question is asked for 3 marks, you can write down the rate of reaction decreases beyond pH 6. Okay, the rate of reaction decreases beyond pH 6. Okay, so you can get 2 marks just for writing that as pH increases, the rate of reaction also increases. The enzyme activity, or you can say, sorry, not enzyme activity, you can write down as pH increases, the rate of reaction also increases, and then the rate of reaction was highest, or rate of reaction was at its peak at pH 6, and beyond pH 6 the rate of reaction decreases. That's it. Only this much is visible in the pattern of this particular graph. If you do not want to if you want to actually include quoted data from the graph, you can write down at pH 6 the rate of reaction was maximum, and it was at pH 6 how much it was? It was 1. Okay, so at pH 6 the rate of reaction was 1 centimeter cube per minute. Okay, so remember that whenever you're using the quoted data from the graph or table, you must write down the unit. Okay, so at pH 6 the rate of reaction was the highest, and it was 1 centimeter cube per minute. The second question is explain the changes in the reaction rate between pH 6 and 8. Now between pH 6 and 8, you have to explain the reason. Now you don't have to describe. Okay, if they ask you to describe, you just have to write down that beyond pH 6, or as pH increases from 6 to 8, the rate of reaction decreases. Okay, this is described. But they are asking you to explain. Now in explain you have to give the reason behind this decrease in rate of reaction. Okay, so you are aware that what could be the reason. The question is asked for 3 marks, remember. Okay, so you can write down that at pH 6 the rate of reaction was highest. Okay, this is because pH 6 is the optimum pH of the enzyme. Okay, or you can say that because pH 6 is the optimum pH of the enzyme, hence the rate of reaction was at its peak. Okay, and next you can write down that for writing this particular point which I've told you just now, that at pH 6 the enzyme activity was highest or at its peak because pH 6 is the optimum pH for enzyme. For writing this you'll get only 1 mark. But see, the question asked here is for 3 marks. So for gaining another 2 more marks, you can write down that after pH 6 the rate of reaction decreases because the enzyme because after optimum pH of the enzyme, when the pH goes beyond optimum pH, the enzyme gets denatured. Okay, the shape of active site does not remain complementary to the shape of substrate molecule. Hence, substrate molecule cannot fit with the active site, and therefore the rate of reaction will decrease. Okay, so I hope you are clear with these 2 points, describe and explain. Whenever a question is asked on the describe, you actually have to write down the pattern of the graph, and you can also quote data from the graph or table. Okay, this is very important. You can also write down any data from the table or graph, but remember, whenever you write down data from the table or graph, you must mention the unit if it is given. Okay, and in case of explain questions, you actually have to give reason behind these changes. Okay, they give reason behind such pattern of the graph. Okay, for example, between pH 6 and pH 8, the graph shows decrease in the rate of reaction. Okay, so that is described because from pH 6 to pH 8, the rate of reaction is decreasing. That is described. Now in explain question, you have to explain the reason behind this particular decrease in rate of reaction between 6 and 8 pH. Okay, and you are aware that the answer is between pH 6 and pH 8, the rate of reaction decreases because pH 6 is the optimum pH of the enzyme. So enzyme works best at pH 6, or the rate of reaction is highest at pH 6. But as pH goes beyond optimum pH of the enzyme gets denatured, or the shape of active site of enzyme changes, and it do not remain complementary to the shape of substrate molecule. Hence, substrate molecule will not be able to fit with the enzyme's active site, and no reaction will take place. Okay, so only these many questions in this particular video. I really hope that you're clear with clear with some of the very important points discussed in this video, like calculating rate of the reaction, plotting of graph, and then how to answer the describe and explain questions. Thank you. Take care.